Well, thank you for this opportunity. My background is as a neurobiologist, evolutionary theorist, and you will see that that's reflected in this presentation, which is titled Inverse Darwinism, Evolution as Thermodynamic Work. It's an effort to provide a uniquely different approach to the problem of explaining how biological information could have evolved from thermodynamic constraints. The argument is based on a slight rethinking of the logic of natural selection that I'm calling inverse Darwinism because of the way it complements the subtractive logic of natural selection with a generative kind of logic. This is also necessary because natural selection theory is effectively agnostic concerning the thermodynamics of living processes, despite the fact that it's implicitly based on it. Competitive elimination of less fit individuals is ultimately due to their relatively reduced ability to access resources critical for maintenance and reproduction. Because of their far from equilibrium constitution, organisms are at constant risk of breakdown. So just breaking even is not sufficient to stay ahead of the second law of thermodynamics. To do this requires the production of excess replacement parts, as well as excess offspring. Both require physical work. So this generative aspect of evolution is intrinsically thermodynamic. Likewise, the generation of new information in evolution is ultimately a thermodynamic process. But how do thermodynamic processes create informational relationships? Darwinian explanations are often framed in inverse engineering terms. When we reverse engineer man-made artifacts, we attempt to discover the functions that they were designed to accomplish. We often also treat evolved structures similarly, asking what function they were selected for. But evolution proceeds by continually repurposing traits. So what some feature was once and most recently evolved to accomplish is seldom going to be informative necessarily about its origin. This is also the case for even the oldest and most conserved features, such as the transcription and translation chemistry shared by all living organisms. In this presentation, I will argue that RNA originally evolved for non-informational functions and only subsequently was recruited for this capacity. And therefore, its ability to inform other molecular processes is something that evolved. This will convert and invert evolutionary sequences that are often suggested by standard Darwinian theories. But before addressing this evolutionary question, we need to first disambiguate three distinct uses of the term information. The most basic I will call structural information at the bottom here. It concerns only the intrinsic physical and statistical features of a communication medium. The second I will call referential information. It additionally concerns the relation between medium properties and something else, something not intrinsic to the medium that it refers to. The third I will call functional information. It additionally concerns the way that what is referred to can be utilized to organize work to produce a specific end. Often, only often only context determines which of these uses is being invoked. In the physical sciences, for example, only the structural sense is being used, but in biology, all are relevant. However, reference, sometimes also described as aboutness, is not an intrinsic property of anything. So an intrinsic property like the replicability of a gene sequence is not enough to make it about anything. The property of informing and recording the chemical thermodynamics of life needs to be something that is extrinsically imposed on something, on a molecule like DNA or RNA, because it's not an intrinsic property of the molecule. To explain how this could have evolved, I will introduce an expansion of evolutionary logic that I will call inverse Darwinism. Inverse Darwinism inverts the Darwinian algorithm, but in only one respect, that is, this respect of selection. Selection is the result of the multiplication of copies of organisms or their parts in a context of resource limitation. In this context, variant copies that are better suited to that context are less likely to perish. But this sort of context is not ubiquitous. In the context of resource surplus, on the other hand, where most variants persist, only the incompatibility with other variant replicas will decrease their persistence, while combinations that are complementary to one another will be favored. Moreover, duplication itself becomes a source of redundancy. This can also minimize the cost of the reduced functionality that might come from maladaptation, and it will enable an increase in variation of the alternatives. To briefly summarize an otherwise complex logic, follow the following. In an inverse Darwinian process, there are 4 phases. First, the duplication of some functional components that will create functional redundancy. This provides comparatively protected context that buffers any negative effects that might result from a degradation of function. In information theory, for example, redundancy compensates for noise. In evolution, however, redundancy produces error tolerance. Second, duplication also enables relatively neutral degenerative effects to accumulate. This includes partial degradation of the original function and redistribution of where or when this function is expressed. Third, by redistribution, potential negative interactions among the variants will be minimized, enabling a higher rate of retention and an increased probability of uncovering complementary interactions. And fourth, complementary interactions will tend to make any loss of autonomous functional abilities less costly. This will further their degeneration until eventually codependence becomes irreversible. Gene duplication provides a simple example of inverse Darwinian logic. The functional redundancy provided by gene duplication relaxes natural selection and allows degenerative effects on a copy to accumulate with minimal reduction of function. In addition, prior functional overlap of the duplicate gene increases the probability of exposing synergistic interaction effects simply by chance. In the case of the evolution of primate color vision shown here, duplication of the green opsin gene, which codes for the light-sensitive protein in the cones of the retina, allowed degeneration of the duplicate gene so that its protein could produce reduced responsiveness to higher wavelength light. This shifted sensitivity more to the red end, so that segregation of now what were separately red and green opsin proteins in distinct cones enabled their non-interference. And then, therefore, there's the separation of their effects in transmission back to the brain. The distinct signals from each differently sensitive cone therefore interacted in the brain to produce what we now experience, we primates, as trichromatic color vision, shown here on the right, compared to dichromatic vision that most other mammals experience. A related effect is also produced by duplication that is external to the organism. An example is provided by the loss of endogenous ascorbic acid production, that is vitamin C, that happens in primates like ourselves. Although most animals possess a gene that produces a protein that catalyzes the production of the antioxidant ascorbic acid, in humans and other monkeys and apes, the homologous gene is non-functional. The redundant availability of dietary ascorbic acid from fruit eating in early primate evolution appears to have enabled the degradation of the gene leading to this process, so we become irreversibly dependent on eating fruit. This dependency additionally shifted natural selection to favor any other adaptation that stabilized this codependence. One likely consequence is the evolution of trichromatic vision in primates that I just described. That aids in the identification of the ripeness of fruit. In what follows, I will use this basic logic of inverse Darwinism to offer a molecular thought experiment that demonstrates how thermodynamic constraints can become offloaded onto a molecular structure, like RNA, for example. This will avoid merely assuming the existence of molecular information, and instead will show how such a molecule might acquire informative properties that it does not originally have. The process I will start with is called autogenesis. It's modeled on a virus structure, but it is not parasitic. This is a molecular system that is constituted by two linked self-organizing chemical processes. One's called reciprocal catalysis, and the other is self-assembly. The two are linked by a side product produced by catalyzation that tends to self-assemble, a molecule that tends to form into a closed container. But in this case, the closed container will tend to capture catalysts that produce it. So if the containment is breached in an environment that includes appropriate catalytic substrates, re-encapsulation of the ensemble of reciprocal catalysts will become much more likely. And as a result, an autogenic virus like this will repair itself, even though it has no RNA or DNA and is not parasitic. The details of autogenic thermodynamics are depicted in these two diagrams. The diagram on the left depicts the two complementary self-organizing processes as large arrows, self-assembly in green and reciprocal catalysis in red. The reciprocal production of each other's requirements and the boundary conditions they need is labeled and indicated by dashed gray arrows. Both self-organizing processes will tend, however, to run to equilibrium and dissipate if they're not linked. This fact is indicated by the dashed black arrows on each, indicating how entropy production leads to their eventual termination. The diagram on the right depicts these processes when linked by a catalytic side product that tends to self-assemble into the container. This yokes the two self-organizing processes together into a higher order synergistic thermodynamic unit. This facilitates the persistence of both self-organizing processes so that they don't run to termination. It prevents their separate descent into this irreversible state and internalizes the potential to preserve the organizational integrity of the whole. This linkage, diagrammed as a yellow double arrow, is effectively a higher order constraint on the constraint producing processes that are codependent in this system. The result is a self-preserving system with distinctive thermodynamic properties. This diagram depicts two cycles of breakup and reconstitution of such a simple autogenic virus. In each cycle, the capsid, the container, is breached in an environment with appropriate substrates, and the release of catalysts enables work to regenerate the inert phase. Notice that the hologenic constraint that holds it all together is preserved despite dissolution, reconstitution, and replacement of all the molecular components over the course of many damage and repair cycles. New molecular substrates and free energy supplied by the environment are constrained by this constraint so that only self-reconstitutive work is performed. The transfer of this constraint from substrate to substrate transfers information that effectively remembers and regulates the autogenic reconstruction process whenever this integrity is breached. To complete the autogenic scenario for explaining how a molecule like DNA could acquire informational properties that it didn't originally have, I need to introduce one other kind of molecule, a nucleotide. I will skip over the still problematic issue of the abiotic synthesis of nucleotides, like ATP, that is assumed by the RNA world theories and their cousins. And I will be assuming that nucleotides can be formed as products of catalysis, as they are in all living organisms today. This scenario is inspired by a curious biological coincidence. Besides their informational functions in contemporary organisms, like their roles in RNA and DNA, nucleotides also serve as the primary vehicles for energy transfer within living cells. Nucleotides like ATP take up and transport energy in the form of high-energy phosphates, shown here as green crosses. Assuming that nucleotides can give this energy off to other molecules, it can modify their functions. But assuming that nucleotides originated for their energetic properties avoids assuming any pre-existing informational or replicative function. However, nucleotides that are energized with high-energy phosphates can also be disruptive in contexts where chemical processes need to remain stable, such as the inert phase of autogenesis. The physicist Freeman Dyson once suggested that by forming into polymers, nucleotides could be stored when their energy conveying function is not relevant. However, a nucleotide polymer, a linearized RNA polymer, for example, can be repurposed for other functions, and those that I want to focus on. For example, one property of a single-stranded RNA polymer is that it will tend to fold into branch-like 3D structures. This is due to the complementary base pair attraction that can spontaneously occur between different regions of the molecule. This creates short double-stranded branches. Non-paired regions will form loops, bumps, and hairpin-like structures connected by these branches. This means that the sequence of nucleotides intrinsically embodies a pattern that is correlated with the 3D configuration that the molecule will tend to assume when it folds. In information theoretic terms, you could say that the sequence shares mutual information with the 3D structure and vice versa. However, this does not make the one information about the other. The correspondence is merely an intrinsic property of the molecule. However, it does provide an affordance that can be utilized for other purposes. Because the 3D branching structure of a single-stranded RNA molecule is a function of complementary nucleotide to nucleotide binding, the replication of complementary RNA sequences depicted on the top will produce a non-identical RNA string that nevertheless will tend to fold into an identical branching structure. But because it is constituted by completely different complementary nucleotides, its loops and unmatched regions will have different chemical properties. So their 3D conformations will be structurally identical, but the two molecules will be chemically distinct in interaction with other molecules like DNA and RNA. The branching geometries of 3D RNA molecules can also provide structures onto which other kinds of molecules, like the globular proteins of the body, can bind via their complementary molecular structure. Specific RNA protein pairings characterize much of the complex molecular machines that are found in cells, such as ribosomes and spliceosomes. Since 3D RNA structure is sequence dependent, particular protein binding patterns on these molecules will thus be correlated with a particular RNA nucleotide sequence. This is a redundancy that can initiate further inverse Darwinian processes. For example, 3D RNA shape can influence the relative proximity and orientations of the molecules that tend to bind to it. This can affect their probabilistic interacting, such as between particularly catalytic interactions between molecules. Since template-based synthesis of an RNA strand, producing a complementary sequence will produce a nearly identical 3D shape, this bias can be replicated, can be inherited, and can be transmitted. While the non-identical chemistry of the unpaired loops will link with different molecular contexts, this will enable non-overlapping distribution of these redundant effects. But if a catalyst tends to bind to a template structure in a pattern of relative proximity that is analogous to their probability of functionally beneficial interactions, indicated on the right by the solid arrows, the probability of non-functional interactions, indicated by dashed lines, can be reduced. In this way, the template effectively functions as a second-order catalyst by lowering the probability of some catalytic interactions and increasing the probability of others. This will favor the persistence of some sequences over others by virtue of sustaining the autogenic system that they are a part of. And it will overcome a classic problem that we see in this kind of combinatorial chemistry, oftentimes called Eigen's cliff or a catastrophe problem, where too many interactions basically derail the relevant interactions. This effectively answers what could be described as the RNA grounding problem. The mutual information shared by globular protein structure and linear DNA nucleotide sequences is mediated by the correlation between RNA 3D branching geometry and linear RNA sequence order. Although this scenario does not address the evolution of the genetic code and protein synthesis, it does demonstrate how the constraints that organize specific molecular interaction patterns can be offloaded onto the linear pattern of monomers in a polymer like RNA. Due to the correlation of RNA 3D structure with the sequence of its nucleotide polymers, information about catalytic and other protein-to-protein interactions can be accessed, preserved, and reproduced in this highly compressed and efficient form. This inverse Darwinian scenario provides a molecular thought experiment demonstrating a plausible evolutionary path whereby a polymer like RNA can be endowed with the property of carrying information about other molecules and their interrelationships. In 9 steps, it is traced the way that constraints on the organization of molecular interactions can be offloaded onto a molecular structure that can serve to regulate, record, and transmit this information in a highly compressed and efficient form. The final step in this sequence at the bottom involves reverse transcription of an RNA sequence onto a DNA sequence. Due to the robust double helix form that DNA takes, it is completely insulated from the dynamical details that it influences. And so offloading onto this robust structure maximizes the fidelity of information storage and transmission, making modern evolution possible, the evolution of all cellular organisms and even viruses in the current form of life. To conclude, the scenario suggested by this approach addresses the molecular evolution that must have taken place prior to the last common ancestor of all cellular life forms, that all use the same basic genetic code for synthesizing proteins. So this scenario only sets the stage for the evolution of the incredibly complex synergistic molecular processes that reverse this logic in order to synthesize proteins. An even more complex inverse Darwinian process must therefore have progressively duplicated, fractionated, and entangled the protein-RNA combinations that characterize contemporary organisms. But as RNA-assisted autogenic damage and repair cycle that is demonstrated here and shown in this loop on the left demonstrates, there are, in effect, a large fraction of this process takes place outside of the containment of the autogenic virus. Because of this, because of being, in a sense, in a shared environment, there will be extensive horizontal exchange and recombination of the RNA and other molecules in a local population of reproducing and repairing autogenic viruses. Such unconstrained combinatorial exchange of RNAs and catalysts will therefore tend to greatly accelerate the effects of the duplication, degeneration, complementation, and synergistic codependence of the inverse Darwinian process. This may help to explain the origins of the incredibly complex molecular codependencies that have given rise to LUCA, the last universal common ancestor of all life on Earth. In this way, the inverse Darwinian perspective can suggest a multitude of new research approaches to some of these most vexing problems about the origins of life. Thank you.